Our lab is kind of split into three, but two of our main topics these days are, one is modeling neurodegenerative diseases. We're also doing some basic epigenetic studies in mouse stem cells, and also a topic which we call paleoepigenetic, a topic which we spearheaded together with Eran Meshorer about 10 years ago. And this is studying epigenetics of ancient genomes, but I'm not going to discuss it today at all because I don't have time. So next time. So today I'll tell you about our efforts to model Huntington's disease, and I'll tell you the why we do it, etc. But before that, there's a birthday which we need to mention. He was born exactly today, over 250 years ago. I guess most of you don't recognize him, but you do recognize his name. His name is Jan Pourquinier. Happy birthday, Jan. Of course, he discovered Pourquinier cells, but many other things. Actually, the cell cytoplasm, the protoplasm, what he called, is also his discovery. He was the first to recognize the compartmentalization. Very fascinating life. Another person I'll just mention briefly is George Huntington, another 19th century scientist. He was a physician. He published only 2 papers during his lifetime. The first one is the Korea Huntington. Talk about impact. So quite impactful.
So not like a God, only one publication.
Yes, maybe the second after God. So Huntington's disease is a late onset neurodegenerative disorder. You can see the incidence is about 7 for 100,000, almost identical for males and females. And it causes degeneration in the striatum, mostly of medium spiny neurons, GABAergic cells. And there's a known mutation since 1993, and the mutation is a CAG expansion inside a gene which is called after the disease, like many others, because we don't know what it's doing. It's called Huntington. And in the normal population, we have somewhere between, normally it's 15 to 20 CAG repeats, but it can be extended up to 39 with no apparent phenotypes. But in the disease, in the patients, you see 39 and above with 100% penetrance. Okay, so if you carry 39 CAG repeats, you're going to die of Huntington. If a little bit less, there's a gray zone. And of course, it's a...
Can I ask a naive question? How come we don't know the function of this protein?
Yeah, it's not that we don't know the function, but it seems to be a multifunctional protein. So there are lots of functions in the literature. There are maybe more than, I would say, up to 10,000 papers that try to describe a function. So you can find it, first of all, in terms of the localization, both in the nucleus and the cytoplasm. And the normal function, you can really find a lot of different things, mostly to do with BDNF secretion, but there's microtubuli, mitochondria, nuclear function. So this is one of those proteins that, there are so many functions that you can really say that we haven't really pinned down the one true function. So it's not that we don't know anything about the function, but we know too much about the function. So this is the case with not only Huntington but many other proteins. Okay, so the disease itself, it's neurodegenerative. They start by chorea, involuntary movements, but then they succumb to the wheelchair, cognitive decline, and ultimately death. If you look at the protein, so the CAG repeat in the DNA is inside exon 1, which is inside the coding region of the protein. So the produced protein actually has an amino acid, an extended amino acid. CAG codes for glutamine, encoded by the letter Q. So you get a polyglutamine protein, or a polyQ, as we call it for short. And Huntington's disease is actually one of 9 such polyQ related disorders. We actually have, it's quite an interesting story, because when we got into it, we thought, okay, we have 9 genes with polyQ. makes sense in the normal population, and when this is extended, we get extended poly-Q and 9 diseases. But when we started investigating, we found more than 50 proteins with poly-Q and only 9 diseases. And we actually, it's a work that was published a couple of years ago, so I'm not going to talk about it today at all, but we found some idea why these 9 diseases, why these 9 genes cause diseases and the others don't. If you're interested, we can talk about it later. But the fact is that these poly-Q proteins, they probably have some kind of misfolding problems. The poly-Q stretch is kind of a sticky wicket. We'll go back to this idea. You get misfolding and aggregation. So if you express in cells a poly-Q extended protein, you get these poly-Q aggregates. Whereas if you just do like 30 Qs or 20 Qs, you don't really get aggregates. So you have this kind of bona fide cellular phenotype, which we were very excited about when we started working on this many years ago, and we didn't anticipate the challenge that I'll show you in a second. Okay, so what do these poly-Q aggregates do in Huntington's? I asked ChatGPT, like we do these days. So this is the common knowledge. ChatGPT says poly-Q aggregates formed by the mutant Huntington protein are generally considered toxic to neurons. It makes sense. They can disrupt normal cellular function and lead to neuronal dysfunction and death. So this is what you compile from the literature today. This is the common knowledge. There is a very clear reverse correlation. So each one of these dots here in this graph is a patient. So if you plot hundreds or thousands of patients worldwide, this is the expanded poly-Q or CAG length, and this is the age of onset. You see there is a very clear reverse correlation. It's almost deterministic. If you have 50 or 60 repeats, I can tell you with quite certainty what age you're going to die. It's really, these people are ticking, bombs, and there's nothing the modern literature can give them. I just talked with Hagai. He said they operated 3 patients, but even DBS doesn't really help them. So what I want to tell you today is that, first of all, Huntington's has an epigenetic component, which kind of brings us to our world. This is just cumulative evidence over the past 15 years or so. But also, importantly, we believe, and not only us, there's again accumulating evidence to suggest that there is a neurodevelopmental component. And this is, I think, very important because this is a deterministic disease. The penetrance is 100%. We know 40 years before that they're going to develop Huntington's and die. Why do we treat them only when they're going, they have 5 years to live? So our idea was to really try to investigate pre-symptomatic. Nomi talked about it last week in regard to Alzheimer's disease. So the idea is really to understand pre-symptomatic molecular alterations using cells, of course, many years before the onset of the disease. And another point which I'm going to kind of allude to as well is there a loss of function component. So Huntington is a dominant disease, okay? So these patients, they have one mutated copy, and that is sufficient to cause the disease. There are very few homozygotes in the world. So by and large, over 98% of the patients are heterozygous, and of course it's a dominant negative disease. So we know there's a gain of function by definition. But the question is whether inside this gain of function phenotype there's also a loss of function because you have one allele that is lost. So this is something that we are able to investigate again using cells. So this is just a few papers that came out in the past several years. Elena Cataneo, one of the leading Huntington's disease researchers in Italy, already commented about chromatin and epigenetics dysfunction in Huntington's disease over 15 years ago, but there are really kind of dozens of papers and reviews. This is just a handful, and we also contributed to this notion with our own understanding, and this is what I mentioned earlier, that actually we found, if you look at the epigenetic structure of those nine genes versus the rest of the 50 that don't cause disease, we actually found that there's a special epigenetic signature for these 9 genes, which we believe could lead to aberrant expansions where the others are more protected. Again, this is something that we published several years ago. We had actually 2 ELSC students, one already in postdoc, and Leah just leaving like today, I don't know, maybe tomorrow. Okay, so and the neurodevelopmental component, again, more cursory in terms of the actual papers that were published, but there's one paper published over a decade ago where they looked at children, and they found that gene-expanded children had significantly lower measures of head circumference. So this is, again, before the onset of the disease. These children are otherwise healthy. It's not juvenile forms of Huntington's, which there are. I'll tell you about it in a second. So these are late onset forms, but you can already see a phenotype very early. Another paper in Brain a few years later showed, again, they studied patients and they showed that the results, a disease burden-dependent functional reorganization of whole brain networks. They did it molecular and functional integration of large networks in the brain. And finally, an important paper that came out in Science just 3 years ago followed something like 15 or 16 aborted fetuses of Huntington, carrying Huntington. So this was it took them a long time to actually collect these samples. But what they found is that they all share this phenotype of the mutant Huntington's shifts neurogenesis toward neuronal lineage. So it's a phenotype of some kind of accelerated neurogenesis in these aborted fetuses that carry the mutation. Again, of course, there is no phenotype. It's not majorly expanded. These are late onset cases, but still there is some kind of neurodevelopmental component. So we decided that we can actually model late onset neurodegenerative diseases using very, very young cells. In fact, 0-age cells, human pluripotent stem cells. These are either induced pluripotent stem cells, which are actually very easy to get these days. You obtain normally skin fibroblasts from healthy people or from patients, but you can also do it from blood and other cell types, whatever you're willing to give. And then you can just reprogram them using just 3 or 4 factors, which are even non-integrating, and then in a matter of a couple of weeks, they reprogram into these iPS cells, very famous. But also, you can use in vitro fertilized embryos. So these are couples that one of the parents have the Huntington's mutation, and they want to have healthy children. So they will undergo what we call elected IVF, in vitro fertilization, not for fertility reasons, but for screening embryos. And then what they do when they do this screening, they actually pluck 1 cell out of the embryo at the 8-cell stage. So this is about 3 days after fertilization. And then they just do a PCR for the Huntington gene. So they screen all the embryos. There's usually with IVF, you get more than a dozen. And then the healthy embryos are either frozen or put back in the womb for pregnancy. And the ones that carry the disease, they can go to labs such as ours. So we also have human embryonic stem cells with Huntington's mutation. Actually, it took a while to locate them in Israel for some reason. I'm not going to get into it now, but just several years ago, in collaboration with Racheli Aiges at Sha'arei Tzedek, we obtained the first lines in Israel. So far we worked with Australian lines, but anyway. Today I'm going to talk about IPSLs from patients. And the reason is that we really wanted very long repeats because we were kind of aiming for real neurodevelopmental phenotypes. So I'll show you what we did. Okay, so once we have the mutated line, we can actually use CRISPR and correct it. So we have what we call isogenic lines, where whenever I say isogenic, I mean that we have two identical lines, one carrying the mutation and one with the same genetic background, just with a corrected mutation, where we introduced 20 CAG repeats, 20 polyQ, so we can really compare these cells head to head. Okay, so how does Huntington's cause disease? So I already mentioned that there is a cellular phenotype, polyQ aggregate. So again, we were very naive, and I saw Wala just come in, and she spent a good 2 years of her life struggling over this problem. We said, okay, it's very easy. Let's take polyQ, GFP, like people have done in HeLa cells or 293 and other cell types, and just express it into our cells, and we'll get phenotypes, polyQ aggregates. But it turns out that human pluripotent stem cells, they're very fussy about this. They don't like to form aggregates. So normally when you transfect the cells, you get this diffuse localization, whereas if you do it like in 293 or HeLa cells, you get much more cells that actually express this polyQ aggregates. So this was frustrating. But so we took two directions. I'll start with actually work by Moria, and Elad later joined, which was to actually. Yes.
When did you exactly transfect your
I'll
the
get
IPS level, or when they are up to differentiate?
Yeah. So this was an IPS level, but later on. We'll differentiate the cells and try again. Okay, so be patient.
I am patient.
Okay, so we took two approaches. One was Moria's PhD, which was to actually go back to the cells and try and label endogenously both the wild type and the mutant HTT proteins with two different fluorescent tags. So we'll be able to monitor and track both endogenous proteins, and the other one is actually enriched for polyQ aggregates. This is Wala's PhD, and I'll go back to it in a few minutes. So we'll start with this. The idea here was to try and, as I said, knock in fluorescent proteins to the wild type and the mutant alleles. Now, it's not an easy task because the two alleles are identical. There are no SNPs to separate them. So we had to do it kind of blindly. We couldn't distinguish between the two alleles. So we took two cassettes, one with M-cherry and one with GFP, with the same CRISPR tagging approach, right? The same guide RNA, and we had to just screen for the ones that will be double tagged. So again, we tried with the undifferentiated cells to FACS sort both GFP and cherry positive clones. It didn't work, and very frustrating, several months of work. But eventually we thought, okay, maybe the expression level is not high enough in undifferentiated cells. And then using microscopy and just sequencing, we were able to identify clones that look like this. So we have CAG wild type. This is the 20 repeat. Fused to M-cherry. We actually used the C-terminal of the protein, so we eliminated the stop. You have an M-cherry in fusion with the Huntington, and we added flag just for purposes of pull down, etc. And then the stop is here, so we have a fusion protein that is red. And then in the same cells, we have the mutant Huntington, which is fused to GFP. So we have dual labeling. And the nice thing that Moriah actually was able to pick a reciprocal clone, where the wild type is now green and the mutant is red. So we can do reciprocal experiments, completely eliminating all issues with antibodies and stuff like that. So we always did the same experiment twice, once with GFP pull down and the other with cherry and vice versa. So we had a very strong system to actually look at what's going on with the two proteins within the same cells. This was the first time, because so far people have actually looked at cell populations where you have the mutant and the wild type versus the wild type and the wild type. But here we have the wild type and the mutant labeled within the same cells. So this is just the controls to show that they do actually co-localize. So the next thing we wanted to differentiate our cells, and what Moriah chose to do is a three-dimensional differentiation into brain organoids. So now it's really an explosion of protocols to not only differentiate the pluripotent stem cells into neurons or specific types of neurons, but also into these three-dimensional brain-like structures, which kind of mimic a fetal brain, if you will. So you can do it for months and months on end. But we eventually decided to work on a 1-month-old organoid. First of all, to look at the very, very early changes that occur, but also from practical reasons, we didn't want every experiment to take 6 or 9 months. They formed these really nice, elaborate brain-like structures. This is just a confocal section through one of those 1-month-old organoids. This is a 5.5 months old organoid. If you zoom in, you can see these nice neurons. This is beta tubulin 3 staining. And this is a zoom out. So you can see MAP2, mature neurons, TAJ1, again, beta tubulin 3, more early neurons, and you get different cell types. We'll go back to it in a second. Also, if you look closely, you can see these are organoids with no GFP and cherry. Maybe I'll just this is a little bit better. Yeah. So here you don't see anything. This is just some autofluorescence. But here you can start seeing cells which actually also produce these aggregates. And if you zoom in, you can see some of these double labeled yellow GFP and cherry aggregates. And it's also nice to see that we actually, these aggregates start forming by the mutant, and then the wild type joins them. So eventually we see them yellow because you have both wild type and mutant, but at some point you see only the mutant and then the wild type. Okay, so this is just the system, but we really wanted to know how the poly-Q expansion affects Huntington's interaction. Because again, we have this system where we have both proteins labeled in the same cells. So the experiment that Moriah did was to separate the wild type and the mutant proteins with IP, with immunoprecipitation, because we can now use antibodies either for GFP or cherry or flag and HA, the tags that we have tagged our wild type and mutant proteins. Because we have the double label, we can do the anti-GFP pull down, and then we take the GFP pellet and then proceed to anti-cherry pull down. So we get both entities, and we can do the reciprocal experiments because we have cherry and GFP tag. This is just the controls. Let's skip that. Okay, so we took pooled organoids. The organoid to organoid, there's quite a large amount of heterogeneity. So to avoid heterogeneity, we pooled together a number of organoids, and then we did this experiment. And we used mass spectrometry to identify the proteins that bind either the wild type or the mutant. So we got a list. I'm not going to bore you. Just highlight one particular protein which we focused on. And this protein is called DNMT3B, or DNA methyltransferase 3B. And this protein, does a very important function during development. It methylates DNA. Of course, we're particularly interested in that because we're interested in DNA methylation and epigenetics in general. And if you go back to development, there is in primordial germ cells, the cells that make the sperm and the egg, there's a particular signature of DNA methylation. When they meet and fertilization takes place, in the first 1 week of development, there is a complete erasure of DNA methylation, and then after implantation, there is a reestablishment of the DNA methylation during development. So this protein, DNMT3B, is the de novo DNA methyltransferase. There are actually two, DNA 3A and 3B, we'll call them for short. And they actually do all the work. They re-establish DNA methylation during development, so they're very active from the blastocysts. They're actually very highly expressed in pluripotent cells because they're kind of waiting for their action in early embryos, but then most of their function is during early development. So this makes sense that we have them in our 1-month-old organoids because this is early development. So what we decided to do is we tried to go for whole genome methylation maps, and this is where lab came into the story. So we did this Illumina and Femu methylation epic array, which actually have 850,000 methylation sites, covering a lot of the interesting CPGs in the genome, not everything, but a good coverage. And you basically, what you do in order to identify the DNA methylation is a method called bisulfite conversion, where you basically apply bisulfite to your cells, to your DNA, and every cytosine will be deaminated into a urine. But if it's methylated, and this is the crown that you see on its head, it's protected from bisulfite, so it will remain a cytosine. And then you could just compare the sequence before and after, and you know whether this cytosine was methylated or not. We applied this to our organoids. Again, we pulled together several organoids, isogenic organoids, and what we're looking at now are organoids with 72Q. 72Q is from a girl that developed Huntington's disease at the age of 19. Okay, so this is a juvenile form, kind of an early onset. It's still late onset. She's 19, but it's considered quite rare, quite dramatic. 72 is a long repeat. So this is kind of to bring the symptoms more towards a juvenile case. And what we found was really major differences. This is just an example of how the data looks like. You have points here that each one of those points is a methylation level. And if you compare the red and the blue, you compare the isogenic cells with and without the mutation. And here in this region, you see that there is a hypomethylation in the mutant. And she did it 3 times. And the repeats are very reproducible. Again, pulled organoids to get rid of heterogeneity. And the bottom line was that we found global DNA demethylation in the disease state. So most of the CPGs, they don't change as we expect, but there's about 10% of those that show decreased demethylation in the organoids and less than significance increase. So this is a chance level. Okay, so we have a 72Q and we have global demethylation. So to see whether this is really poly-Q dependent, we took another isogenic cell. This is just to show the 3 experiments, the controls, the mutants, they're a bit more heterogeneous. And here you can see this decrease globally. If you look at the DNA methylation, this is a classic DNA methylation profile because DNA methylation is biphasic, it's either 0 or 1 because it's binary. Now, interestingly, I told you there are two DNA methyltransferases, 3A and 3B. Now, it's very difficult to look, impossible in fact, to do what we call ChIP-seq. I'll tell you about it later. But to look at their actual binding. But each of these two proteins, they have a different motif that they recognize. Very similar, CG of course, they both recognize CG, but either a CT afterwards is for the A or a GI afterwards is for the B. So we were able to actually, Elad was able to compare these signatures between our data and what we expect in terms of the 3A, 3B. And if you do that, it's really striking to see that we have, this is a p-value of significance. You see that all our data are kind of pointing at a function for 3B. And 3A seems to be not playing a role here at all. So it seems that what we found is really 3B dependent and not related to 3A. Okay, so we worked with so far with a 72 somewhere here. This is a 19-year-old patient. So I would say one of those circles right here. But then we decided to go for an infantile case. These are very rare. In fact, this the one that we decided to go with is 180Q. This is the worst case that for which iPS cells are available. The worst case that was ever reported is about 200, but this is again the worst case that was ever reported with available iPS cells. So we repeated the entire experiment. So it's the age of onset for this patient was 6 years old. A 6-year-old male, for which, as I said, iPS cells are available, and we have the isogenic cells. So we repeated the experiment, and lo and behold, this time we found more than 30% of the CpGs are demethylated. Really global and robust hypomethylation in the disease cells. Again, if you compare this decrease that we saw earlier, this was a 10%. Here we see this 30% decrease in the 180Q. And again, we can go back and test the signature of the 3A, 3B. Is this random? No, it's really 3B related. Again, you can see this very, very, very significant p-value for the 3B signature, suggesting that this is really 3B related. Now, DNA methylation gives us another really nice opportunity, which is something that gained a lot of interest in recent years, and it's called the epigenetic clock, developed by Steve Horvath in Stanford. And this is basically, we can sample individuals and know your age with very good accuracy, just by a signature of a few hundred DNA methylation genes or positions in the genome. Some of them get hypermethylated, some of them get hypomethylated, but they change over time. So this is a really, really nice system to know your biological age. And in some diseases, this age goes faster. And actually, you can even, if you live a very healthy lifestyle, you can be younger than your actual age. But overall, you'll fall on this nice line. So now we have this, late onset age-related diseases. We wanted to see whether we can already in these organoids, 1 month old, right, very, very early on, we can see a difference in this epigenetic age. And this is really really interesting surprise. Here you can see the random age, it's 1, that we assign to these wild type cells or corrected cells. This is the 72Q pair, okay? So we get about a 20% increase in the epigenetic age in these 72 extended poly-Q cells. When we look at the 180Q pair, we see about a 50% increase. And these two have the same epigenetic age, and they come from different genders. This is a 19-year-old female, 6-year-old male. Of course, they're both one month old organoids now. Okay, so their age is reset. but they show the same epigenetic age. There is no reason for it unless this system works. So it's really nice to see that it works, and in the 180 you get this really increased epigenetic age already very, very early on. So I'm really trying to convince you that there are molecular manifestations that are global, are real, are reproducible in pluripotent stem cell derived organoids and probably in ES cells as well. So what about gene expression? Of course, we have these organoids. We already extracted RNA, DNA, so we wanted to look at gene expression, so we repeated this with RNA sequencing. And in order to also add this loss of function dimension, which I told you earlier, Moria also generated knockout clones for Huntington. So again, she used CRISPR, but this time just to eliminate the entire 5' end of the gene. She got rid of the promoter and the first exon. And then she got clones which are completely knocked out for Huntington. So we have pluripotent stem cells with no Huntington versus the ones with wild-type Huntingtons, and then we're going to look at the ones that have disease versus the isogenic controls. So first thing that we saw that in undifferentiated cells, we see very little changes in gene expression. You see these volcano plots here, almost nothing. But when you now differentiate the cells into 1-month-old organoids, you start seeing a lot of changes, hundreds to thousands of genes that get de-expressed, either upregulated or downregulated. You see these blue and red dots, which you don't see in undifferentiated cells. Now, to cut a long story short, what we looked at are modules which share the same pattern between the isogenic disease cells and the isogenic knockout. For example, cluster 2, where the changes, if you just compare the blue and the yellow, the blue are the controls and the yellow are either the mutants or the knockouts, show the same trend. This, for us, is the loss of function, right? The rest is a gain of function because you see them only in the disease state, not in the knockout. But this particular module is what we consider it to be a loss of function phenotype. And if you do the gene ontology analysis of these networks, you see that they're all related to neuronal development. Neurons, synapse, axon development, etc. So we wanted to see the cellular composition of these organoids, and to do this, we teamed up with Oren Ram and Shue in his lab, and we did single-cell RNA sequencing. Again, Nomi talked about it a lot, also Chaya did, and we compared the cellular composition. So this all work was done by a lab, or the bioinformatic work. And first of all, you can actually identify particular cell types, late neuronal progenitors, early neuronal progenitors, neurons, etc. And then you can overlap the disease cells with your isogenic controls, and you quantify each cellular population. And what we found was this really remarkable increased neurogenesis phenotype that we see in the disease state. So more late neuronal progenitor cells and more neurons. You can see these red bars in expense of early neuron progenitors. So just like we saw in this science paper that came out 3 years ago, where they saw in these aborted fetuses increased neurogenesis, we already see it in 1-month-old organoids. So we can actually really model this using pluripotent cells. So we actually believe that not only can we model this is due to a loss of function phenotype, because we also see it in the knockout cells. We see the signature. So we actually have a gain of function related phenotype, which we believe has to do with the DNMT3B, what I told you about, and a loss of function, which is due to the loss of a functional protein. And the model that we suggest for DNMT3B is actually that because we found it to be exclusively associated with the mutant Huntington, we think that the mutant, when it's polyQ expanded, the polyQ starts to sequester all these proteins because it's like a sticky wicket, and then you get sequestration of DNMT3B away from DNA, where it's supposed to do its job. So the longer the repeat you have, the more DNMT3B you sequester, and the less efficient it is to DNA methylate, and therefore you get this global DNA demethylation, leading to this demethylation, epigenetic regulation. We also think it might have to do with the transcriptional variability, which we published a few years ago.
Question?
Yes, please.
You name the clusters after you see them?
Yes.
After you see them, or do you
No, after. Yes, we do an unsupervised clustering. We see what they are, and then we apply just gene ontology just to see what these groups of genes are. Sometimes you have, if it's completely random, you're not going to get any signature, but you can, usually you get something that is meaningful because almost nothing is random, at least in disease states. Okay, so quickly I want to show you Wala's efforts in her PhD. I told you earlier that we took two approaches because we saw no poly-Q aggregates in the disease cells, and so far we really kind of bypassed the aggregate problem. Just we label the poly-Q and the wild-type proteins, and we just molecularly analyze them. But from a cell biological point of view, we didn't see aggregates. So this was really Wala's challenge, and the idea was to try and enrich the population for poly-Q aggregates. And when Wala started working on this problem, she found this paper that was published now more than 10 years ago from a group in Australia, where they basically tweaked the parameters of the FACS in order to find this population. It could be a rare population, but you can enrich for this population that have inclusion bodies or aggregates or whatnot. Okay, so we try to apply that. What I did was to express this GFP fused to 105 Qs inside stem cells, and what we call an exogenous system. But we also wanted, this is a protein that we ectopically express, right? We force our cells to express, but they don't normally have this 105Q. So we also took our, this is the control with 18 CAG. I'll go back to it in a second. I actually labeled it endogenous, but it's not the case. This is the control, a GFP with 18 Qs, which is not supposed to form aggregates. So when you express this 105Q, you don't get aggregates. So then we decided to differentiate the cells into early neurons or neuron progenitor cells, and then try to perform this experiment. And this is exactly what Wala did. So using this pulse A method, after several rounds of you first start by collecting this homeopathic population, which you hardly see, but eventually Wala was able to really collect a nice population of poly-Q positive cells until she eventually got 100% of the cells showing by microscopy at least one nice bona fide poly-Q aggregate. So this is really nice. And now we can actually compare cells with and without aggregates. And I should actually mention that these cells are still dividing. They're neuroprogenitor cells, but they're dividing. So when a cell with an aggregate divides, the aggregate doesn't divide. So by definition, you're going to have two daughter cells, one with an aggregate and one without an aggregate. So when you start with 100% aggregates, after 1 or 2 days, you're going to have a mixed population with and without aggregates, cells that are sister cells growing in the same plate, in the same conditions, in the same incubator, same everything, right? They're sisters, and now you can compare really apples to apples. So it's a very powerful system. So So when you grow these cells, you can now again sort the ones that are poly-Q positive, the ones that are poly-Q negative, and compare them for RNA sequencing, etc., all sort of molecular differences. Okay, so this is the system I was telling you about, cells that topically express 105Q. So the first thing that we found that they're very identical, like you expect from sister cells, very few changes in gene expression. However, we do get a very clear signature, very reproducible, of a few dozen genes, and you can see how reproducible it is. These are 3 biological repeats, and here you can see the genes. We're going to focus on 1 or 2 later. But also, this is the other system I started to tell you about earlier. So ectopically, we express the 105Q. But now we wanted cells that have the endogenous aggregate. So Wallah took these 180Q cells that I told you about earlier, right, with a 6-year-old phenotype, and she ectopically expressed a wild-type version of GFP, poly-Q18. This poly-Q GFP with 18 Qs doesn't form aggregates. But if there are existing aggregates, it will join the existing aggregate. And then you can actually visualize these aggregates using this ectopic expression. And again, she used pulse A for several rounds until she enriched, and we repeated the same experiment, and we have another signature of these genes. And remarkably, again, very few genes, 50 here, 80 here, 60 here, but out of them, 37 show the exact same pattern. So it's really a very, very strong enrichment in these two, again, completely different systems. So it's a clear poly-Q aggregate-related molecular response. And what we saw is a neuroinflammation signature. So some of these genes are cytokines, IL-8, etc. And this is just to validate. This is IL-8 expression. This is one of the genes that we identified. Again, you can see both in the 180 and the 105, we see this increase. And it's a secreted cytokine. Is it really secreted from these cells? The answer is yes. You can see here a ELISA plate, and you can see that they not only express IL-8, but they also secrete it. Okay, so this is a slide I promised for Idan, but Idan is not here. He said he will come. He wanted a brief introduction about gene regulation and enhancers and promoters, so I'll go over it briefly. We really wanted to know which is the transcription factor that is responsible for this increase in neuroinflammation genes. So transcription factors bind regulatory elements and either suppress or activate, usually activate in eukaryotes, gene expression. Okay, so we really wanted to know the binding pattern of, first of all, what to explore and what is the binding pattern. So we looked at these 37 genes, but first, Wallah is this CHIA analysis. It's a chip enrichment analysis. So you basically look at everything in the literature that resembles, looked at all the binding proteins of everything that's available, and looks at what binds these genes that we have overexpression of, kind of to identify potential transcription factors that could be mediating these effects. And 2 of these were in our data set, ATF3 and FOSL1, ATF3 being the top gene and also one of our top overexpressed targets. So it's actually a very well-studied protein. It's an ER stress-related protein activating transcription factor 3, small protein, nice to work with. It belongs to this Krebs family of transcription factors and usually an activator. And nicely, it's really, as of course we found it in our cells that express, that show the poly-Q, but if you just look simply by immunostaining, you can see that cells with poly-Q have high levels of ATF3 and vice versa. So there's really a nice correlation between ATF3 expression and poly-Q aggregates. So we wanted to know which genes in our cells are bound by ATF3. We went back to our system, and voila, again collected cells with and without aggregates. She needed a lot of cells. This took weeks and weeks to collect enough, and then we did ChIP-seq. What you do with ChIP-seq, you fix the DNA-protein interaction using some fixative, formaldehyde or something, and let's just assume that these yellow ones are ATF3, and then you basically come with everything with ATF3 antibodies. So you wash out everything else, you're left only with ATF3 bound to your DNA, and then you release the DNA and you sequence it. So what you get are maps of everything in the genome that ATF3 is bound to, because every peak that you see is actually made of a lot of sequences that accumulate on this particular region. So this is ChIP-seq for ATF3. So if we look at IL-8, for example, this is the RNA sequencing. You see that it's expressed only in aggregate plus cells, like I told you earlier, and no expression. This is one of the induced genes. And now if you look at ATF3 ChIP-seq, you see that it is really only bound in the promoter of IL-8 only in aggregate plus cells. And this was true for other cytokines such as IL-6 receptor. You can see here an enhancer that is bound only in the aggregate plus cells, and etc. So is ATF3 important for aggregate formation? Voila, went and generated an ATF3 knockout in her cells, in the NPCs. And if you go and repeat this IL-8 expression experiment in ATF3, so these are wild-type cells, and in the absence of ATF3, there is no induction of these neuroinflammation genes. So ATF3 is required for this induction of genes, and the key experiment is when she now went back to the cells and did all these pulse-A, sequential pulse-A experiment to enrich for this population of poly-Q containing cells. This was really remarkable because the ATF3 cells remain here, the knockout cells remain here. So there are two types of knockout, and I'm not going into it because of lack of time, but these are the wild-type cells with ATF3++, and these are the ATF3-. So she enriches and enriches and enriches, but no poly-Q containing cells come out. They seem to be incapable of producing poly-Q aggregate, these cells. So here is an ATF3 minus cell, no poly-Q aggregate, ATF3 plus cell in yellow with a poly-Q aggregate. And actually, again, I don't have really time, but our preliminary experiments also suggest that, of course, ChatGPT told us in the beginning of my lecture that poly-Q aggregates are probably detrimental to cells. But what we find is that, in fact, quite the opposite. Okay, so when we now try to poison the cells, we get that cells with aggregates are actually, they cope better with tunicamycin or ER stresses that you apply to the cells. So it seems to be a defense mechanism, not something that will lead eventually to cell death. And we can discuss. I just want to end with this really exciting mouse model that we developed. So Walla went on and generated ATF3 knockout mouse that has no phenotype, and this we knew because someone else already published an ATF3 knockout mouse. But we crossed it with an HD mouse. So these HD mice, they have 70 cues, 70 something repeats, and they succumb to motor defects at around 4 months, between 3 and 4 months of age. And then we have now in the background of ATF3 knockout. And what we found, so this is a grip test. This is a wild-type mouse. So no problem for a wild-type mouse to grip this bar. This is a HD mouse with ATF3. So it struggles. You can see that it's struggling. It can't really go like from one side to the other like the nice wild-type mouse, and eventually it will fall. But when you cross it with ATF3, the phenotype starts much earlier. So and they start more or less the same, and then they succumb, and so if you look at, okay, so this is many trials that we did. This is the grip test, ATF3. They grip less. These are the wild type, and this is the ATF3++, and this is a rotor-road. So at some point they start to deteriorate. These already, all the R61, all the HD mouse, they do much worse than the wild type, right? But the ATF3 seems to be deteriorating at a much faster pace. Okay, so HD has a neurodevelopmental component. I hope I convinced you. There's also a loss of function component. We found that DNA methylation is globally altered in HD organoids. Aggregate plus neural progenitor cells express in secrete cytokines. This is a really aggregate-specific molecular phenotype. ATF3 is activating these genes. ATF3 knockout NPCs cannot form polyQ aggregates. We're still checking this in mice. And these ATF3 knockout HD mice show earlier deterioration. And perhaps polyQ aggregates may be protective. After all, we're still testing this. So this is our lab in a dish, and that's it. So I mentioned Moriah's and Elad's story in the first part, and then voila, in the second part, Matan did the heterogeneity, which I didn't really talk about today, and I'll be able to take questions at those times. Hagai, sorry.
Why is triad?
Why neurons? So
No, okay, why not cerebellum? Take another structure.
why brain? Well, if you're asking two specific questions. Huntington, no, Hagai, seriously. Huntington is expressed at all developmental stages in all cells in our body. It's ubiquitously expressed. Why are only the guys? I've been asking this ChatGPT for like months now. I'm trying to tweak the question that he will read papers and no, he has no idea. Nobody has an idea. There are some hypotheses why this is the case.
What is your report?
I'll tell you more. We don't have one. In the 9 polyQ disorders, completely different genes are affected. One is a ubiquity ligase, one is something to do with the cytoplasm. They all share the polyQ, so we'd expect at least if it's a dominant phenotype, they're going to kill the same cells. No, half of those genes, they're spinocerebellar ataxias. This one is in the striatum. I have no idea.
But that's the reason I ask why not cerebellum.
We're also working on Machado Joseph disease, spinocerebellar ataxia type 3, and now we started doing modeling for type 6. We have no idea. No idea. Yeah, it's fine.
When you perform your gain of function, you showed us that you have premature aging. Okay? And when you do the loss of function, you have premature depletion of neuronal precursors and premature neurogenesis. But what happens to the other event? Namely, what happens to neurogenesis in the gain of function and to aging in the loss of function?
Yes, so what I think it's a temporarily different event. So the first thing that you find probably is a loss of function phenotypes. It's something to do with accelerated neurogenesis, changes, but I think development is so robust. DNA demethylation is that it has a way to overcome these deficits. And later, you accumulate the gain of function. The gain of function is the late event, but we have early loss of function events. This is how we say, I think these are temporarily distinct events.
Yeah, but if you have premature neurogenesis, you're depleting a pool of progenitors. This would account for less neurons in the brain or in the striatum of these animals, isn't it? Neurons do not regenerate if you don't have precursors.
Yes and no. So again, this science paper that I mentioned is in humans, right? Not in animals, in real humans. And we know that they live up to 40 years of age as normal human beings.
So the brain
There's
is
no
not-
apparent phenotype.
The brain is not needed as such.
I think there's
You
a
can
way.
live
You
with
can
half-
be through, can you see?
You can
Well-
live with half the cohort of neurons and still be okay.
I didn't say that. I said that there's a way for development to overcome early neurodevelopmental defects. That's what happens. There is a
I don't understand. No, do you? I mean,
so if you look at the circumference of the head, it's a different. Okay, so it adds up, right? Maybe there is some kind of neurodevelopmental defect which is not corrected, which just doesn't reflect a real bona fide phenotype because these people appear normal. Okay, but there is something there. Yeah.
Just to go with the first question, autism, epilepsy, all kind of things
Yeah.
like that are related, as far as I understand, to early neurodevelopmental issues.
Right.
You showed us that these cells have huge amount of different things relative to normals. So how come these people are not do not show autism, epilepsy, and things like that? I mean, why do you have small defects, small problems? A few genes get bad, and you have autism, but your neurons with all of these defects.
Yeah. So. Of course, it depends what the mutation is, right? So normally in autism spectrum disorders and epilepsy, we have a loss of function phenotype. These patients are usually heterozygous for a specific genes. By the way, many of them are chromatin and epigenetic modifiers. And you have a clear loss of function phenotype that leads to all these changes. Here, I think the loss of function that we see is much more minor than a bona fide loss of an allele, right? This is what you get in heterozygous for neurodevelopmental defects. We are able to see this because we're doing everything you do with a system that is not isogenic, that you don't really compare cells with and without the disease, you hardly see anything. Even with gene expression, when people try to look at all the changes in gene expression from patients and mice, you hardly get anything that is above noise. So it's really, and actually, so going back to this question, the first study that we published was exactly to look at isogenic cells in Huntington's and look at variability in gene expression, a very subtle phenotype. And we had subtle phenotypes in them, but we said, okay, let's take a look at one of those. We took cells that are heterozygous for CHD8, okay, with one allele. Only one allele is mutated. This is one of those genes that causes, with very high penetrance, autism spectrum disorder. It's one of the top SFARI genes. The phenotype, variability phenotypes, was 10 times larger than what we saw in the So I think this makes sense. Yeah.
What conclusions can you come to? We've got this global demethylation.
Yeah.
The fact that people are there even surviving at all.
Yeah, well, maybe DNA methylation is not that important.
Well, it's compensatory.
Yeah, I think it's probably compensatory mechanism. Maybe it's just slow, and later on, the DNMT3, there's going to be enough levels to compensate for all these. I don't know. But if you look at gene expression, it's a significant. It's not that all these promoters get, start to scream expression, but there is a very significant correlation between hypomethylation and increased expression. So there is a molecular phenotype, but again, I think that it's probably not that, development is very robust, so it has a way to overcome these molecular differences.
Have you tried to graft your brain organoids and to follow the fate of
No.
your mutated cells? That would be very nice. Or alternatively, just keep your brain organoids for 5 months, okay, or something like that, quite not economical experiment.
Right.
And then see, because people are able, obviously, to see folding and to see migration along radial glia and stuff like that, and better characterize the cellular basis of your defect.
So you want to extend PhD from 6 years to 16? Yes.
When you
I actually
get-
discussed it with Omer Revach in the Hopper to transform these organoids, and we probably will. There is a grant pending that if we get everyone, but it's really, really, time consuming. It's for us. Good. Excellent experiment.
So is this an expansion disease?
Yes, yes.
Okay, is anything done to avoid the expansion?
Yes, so actually there was a trial which started like a very promising trial, siRNAs. You know siRNA, right, Mona? To try, so the idea was that siRNA are not efficient enough to actually deplete both alleles, and because the expansion has much more CAG repeat, it will be much more effective on the mutated allele, and therefore it will not harm the wild type, and this way you can really reduce the levels of the rogue protein. And in phase 1, they found, so they took CSF samples from the patients, and they found that they really decreased the levels of the protein. They did a phase 2, which seemed to be very promising. Some of the patients actually reported, health benefits of the study, and phase 3 was aborted in the middle. I don't know, maybe Hagai knows
No,
more.
but it was a so big disappointment.
Yeah, I know, the disappointment was-
Everybody came to the patient,
Yeah.
phase 2 is so
Yeah.
successful. We are sure that
Yes.
phase 3, and then next year,
Yeah.
okay, so it is not clear what was happening.
I still don't know.
In
I mean-
phase 2, but
Yeah.
the disappointment.
Yeah, the disappointment was huge disappointment, because they were able to really recruit a lot of patients, and it was really promising, and they didn't even complete phase 3.
Yeah.
It was aborted. We don't even know what's going on there still. It just last year.
Yeah, it was wow. Because patients were told that soon we are.
Yeah, it was a very major setback. This was one of the attempts. I guess if CRISPR manages to be more effective, maybe it will. Now, with CRISPR, you cannot target only this, so it again has to be some kind of dose dependence, but it's probable, because when you target your mutant allele, you're going to target your wild type. So not an easy task. We actually have a patent now for ATF3 IL-8 related pathway. So if we manage to get the disease sooner in mice, we might be able to also postpone the disease if we somehow manage to overexpress ATF3 in this patient. So, all right. איזה כבוד.
רוצים להרחיב את הדעת, עכשיו יש צגות.
כרגיל ניסית יותר מדי, אבל הצלחתי התאמצתי, לא שהיה קל, אבל רצתי.
יש לי מלא שאלות, כמה טכניות. קודם כל, אם הם נכנסים לאגרגט, גם הווייטד וגם האלמנטד, עד כמה אתה נקי כשאתה עושה פול דאון ואתה יודע שאתה לא מביא אותו?
אני חושב שאין לנו אגרגטים.
יאללה, תשובה ערן בעיה.
לא בכלל, אבל כי כשאתה מסתכל על ה-one month old organoids, האגרגטים יש לך איזה 1% מהחיים.
לא, כי שמתי לב שבמודל שציירת, אז תמיד הם נפרדים.
כי ב-one month old organoids, וגם ב-5.5 month old organoids,
אז מה זה הפוסה האלה שאתה רואה?
לא, אני ראיתי לך, אבל אני אומר לך, זה אחד מ-100 תאים בערך. בפולס A, אנחנו עושים מערכת שהיא 100%. מגיעים לזה וזה, אבל זה...
איך זה? למה זה ככה?
כי יש אגרגטים מגדילים. נגיד שאחד ל10,000 תאים. אני לוקח 1,000,000 תאים, מוציא את אלה עם האגרגטים.
בתהליך אתה מקבל עוד?
בפולס A לא, אבל אני שולף רק אותם. ואז אני מגדל את הזה, שולף רק אותם, יהיה לי אחר כך 20%.
כמה זמן זה בין לבין?
3 ימים אנחנו עושים בדרך כלל, ואז חלק מהם מאבדים את האגרגטים, אבל אז
אבל
בפעם
אתה
הבאה,
מנקה אותם.
אני, כן, מנקה.
וכשאתה עושה דבר כזה, אז אתה עושה פול דאון, אתה מקבל גם את הווייטד?
שם אני לא עושה.
לא עושה.
שם אני לא עושה.
דרך אגב, המכשיר החדש פותח את כל הבעיה.
לא, אבל שם אין לי בכלל את הדאבל אייבל, זה כאילו מערכות נפרדות.
המכשיר, אתה מכיר את המכשיר החדש שהוא משלב...
שעושה תמונה.
עם אינסטרים, עם סורטים. לא, עם אינסטרים הוא עושה לך תמונה, זה אנליזר. אבל עכשיו יש מכשיר חדש, חבר'ה בהיידלברג פיתחו, אבל כבר מוכרים אותו ב-BD. אני יודע כי מצבם קצת להוציא הצעה. לא גם זה עם אינסטרים.
אבל כמה זמן זה ייקח?
זה מבוסס על AI Image Analysis, אתה עושה טריינינג על סט מסוים, ואז זה מאוד מהר. זאת אומרת, זה לא מהר כמו פקס רגיל, אבל זה עדיין. זאת אומרת, הוא רץ עוד קצת בצנרת כדי להספיק לעשות את האנליזה, ואז הוא מפריד אותו בסטריק.
הוא גם לא, הוא באמת, טוב, יפה.
אני חושב שהעבודה הראשונה שפרסמו היה משהו אימונולוגי, חיידקים ותאים או משהו, אבל זה עובד, אנשים אומרים שזה עובד גם אצל זה לא רע. ואז זה פותח, אתה כאילו יכול לקחת ישר מתוך האוכלוסייה שלך. לא יודע, האמת שהצטרפתי לזה, כי אני עושה כל מיני דברים, אני גם, יש לי פרויקט שאני מנסה עם פקס לעקוף דברים שאני עושה אנליזה באינסטרים, בלי בעיה, כל מיני טריקים כאלה לעשות דברים, אז אם זה יעבוד, זה נחמד. בקיצור, ספר לך.
אז השתעשענו קצת עם אינסטרים, רק כדי לצלם תאים עם אגרגט, אבל...
לא, אבל הוא יכול לתת לך, נגיד, אינפורמציה יותר עמוקה של מספר אגרגטים.
נכון, אבל...
ויש לו חיסרון כי הוא היה בפלורסנט, הוא לא... זה מה שמסתתר לך מאחורי גרעין.
זה בדיוק העניין, שלא רק שהוא היה בפלורסנט, זה גם הרזולוציה, אני לא... אתה יודע, הרזולוציה לא... אז קצת ויתרנו על זה, כי זה לא באמת נתן לנו את האינפורמציה שהיינו.
אז אנחנו עושים תאים אפיטליאליים שעושים טרנספוזיס תאים איגונים. אז אנחנו רואים תא בתוך תא.
מה הם עושים?
הם עוברים דרך התאים האפיטליים, הם עושים טרנסמיגריישן דרך אפיטל, בקורד פלקסוס. אז אתה לא יכול לראות את זה בפקס בשום צורה.
איזה מערכת?
בעכברים.
אבל זה in vivo.
לא, אנחנו רוצים קורד פלקסוס מפרקים את זה CSL, ואז אנחנו יכולים להימצא כדי לראות את התאים. אז עוד פעם, בפקס ניסינו כל מיני טריקים. פה אתה רואה את זה. מה רוצה לשאול אותך? הפנוטיפ של ה-ATF קשור למטילציה? כאילו מהבית ה... אין לו מוטיב של מטילציה, הוא לא חושב למטילציה.
קשור, כמו שאמרתי, אנחנו חושבים שהמטילציה היא קשורה
ל-DNT. לא, אבל השאלה אם ה-DNT הוא upstream ל-ATF. כי אם אתה משנה את הדגם מטילציה ו-ATF קושר פרפרבלי, אתה יכול לראות את זה בדאטה שלך, אני לא יודע אם זה ידוע עליו או לא.
לא, אני יכול להסתכל על DNA מטילציה באחורים, מה שאני עושה.
כן. הדבר האחרון שלו זה לשאול לך שהוא...
כאילו הם מתפתחים נורמליים.
כן. הדבר האחרון שלו זה שהוא קצת מוזר. אם אתה לוקח בן אדם מאוד מאוד זקן, אבל בריא.
כן, התשובה היא כן. הוא ימות מהנטינגטון.
אתה רוצה אגרגטים?
הבן אדם ימות מהנטינגטון אם הוא יוכל מספיק זמן.
כל
האוכלוסייה...
בן אדם שהוא לא הנטינגטון גנטי. שיש לו normal repeats. אתה מסתכל עליו, על המוח שלו, כשהוא מאוד מאוד זקן. אתה תמצא אגרגטים?
לא, אני חושב שגם האגרגטים האלה באינוויבו בחולים הם די חמקמקים. זה לא כמו בלואי בודי.
אתה תאהב את הרעיון שלי.
כן.
מה אם זה וגם דברים אחרים זה בעצם פרוגרמי? בגלל ההזדקנות. נגיד שמה שזה עושה זה דוחף הזדקנות. וכל האגרגטים האנשים רודפים אחרי הדברים האלה, זה ביי פרודקט.
זה ביי.
אז אתה בעצם עושה הזדקנות.
השאלה אבל,